Welcome to the Channelchek Takeaway Series. This program is provided for informational purposes only, and none of its content should be interpreted as a recommendation or solicitation to buy or sell securities. Investment decisions must be made by you or with the support of your financial advisor. A complete disclosure statement follows each presentation. PDS Biotechnology, NASDAQ ticker symbol PDSB, is a clinical stage immunotherapy company developing a growing pipeline of targeted cancer and infectious disease immunotherapies based on their proprietary Versamune and Infectimune T-cell activating technology platforms. Joining us today is PDS Biotech President and CEO Frank Bedu-Addo. Following a discussion of the company pipeline, Frank is joined by Noble Senior Research Analyst Robert LeBoyer for a Q&A. And now, here's Robert and Frank.
With me here today is Dr. Frank Bedu-Addo, President and CEO of PDS Biotech. The biotechnology industry has gone through a long decline, probably the longest since biotechnology companies have started as publicly traded entities. In the past year, though, PDS Biotech has had data presentations and its stock price has reacted, separating it from most of the peers in its market cap range. Dr. Bedu-Addo, could you tell us what has changed for the company in the last year and how you see the coming year, both for investors and for the company?
Well, Robert, thank you very much. I would like to start by thanking you and the team at Noble Capital Markets for the opportunity to participate in this virtual takeaways event. So just to talk about all what's happened with the company to date, and also together with J.P.Morgan at the J.P.Morgan conference, it was really good to be back at J.P.Morgan after what feels like a really long absence. I felt that although it was not as heavily attended as in years past, it was still very well attended. For companies like PDS Biotech, who are in the clinical development phase, the focus was really to have quality meetings with prospective investors, including institutional investors, and on the business development side, to have introductory meetings with prospective partners or to further discussions towards a potential deal. It's interesting the question you asked, because with PDS Biotech, over the last 3.5 years since we went public, the types of discussions we've been having with prospective investors has changed quite dramatically. Right, in 2019, there was a lot of skepticism around the technology and our ability to translate the outstanding preclinical results to humans. Right, by 2021, with promising clinical data beginning to emerge, the discussions became more along the lines of, okay, this is promising data, but we haven't seen this in enough patients yet. Right, and then by late 2022, as we began to show consistent data across multiple trials, the discussions evolved to having greater focus on the design of our registrational trials and what the regulatory pathway could be to get our products to commercialization and how much that would cost. Right, so as you mentioned, 2022 was a tough year for biotech overall, and we saw significant market cap declines across board, especially for small and micro-cap biotech companies. It's interesting also because if you look at our charts from late 2021 through the third quarter of 2022, our charts look practically identical to all our peers. The data we presented at ASCO in June of last year on 2 of our programs, despite the strong data, seemed to have very little impact on investor sentiment. However, by the fourth quarter, once the MD Anderson Cancer Center data in cervical cancer came out at SITC in November, coupled with their biomarker data, and also the National Cancer Institute biomarker data was also presented at the same conference. I think the strength of the immune correlate data generated independently by both institutions started to give credence to the strong PDS-0101 clinical data that had been presented earlier in the year, as well as the mechanism of action. Investors were also beginning to see compelling data across multiple trials, including the cervical cancer trial where PDS-0101 was not combined with any other immunotherapy. So in this trial, all the immune activation and induction of the potent multifunctional tumor-infiltrating killer T cells had to have come from PDS-0101. We then also presented additional durability data and overall survival on the triple combination, which was again very compelling data showing how long these patients were continuing to survive. And then we started to see clear market cap appreciation and dramatic separation from our peer group on the charts, right? So as you went to the end of 2022 and into very early in January, we saw that significant appreciation in market cap, but this appreciation was clearly data-driven, and we continue to progress these programs through development, and hopefully at least one or two of them into registrational trials sometime this year. And I'll be happy to walk through some of the data.
Could you just explain what each trial was and the combinations that you're using, and then the results compared with what would have been expected historically or from standard treatment?
Yeah, so let's do that. So our lead program, just to give the viewers an introduction, so PDS-0101 is an HPV cancer immunotherapy. It's developed to treat cancers caused by HPV, such as anal cancer, cervical, head and neck, penile, vaginal, and vulvar cancers. By combining Versamune with protein fragments from the HPV-16 protein, the right type of HPV-16 recognizing T cells are induced in the patient's body, which then infiltrate the patient's tumors and have the ability to shrink and kill the tumors. All right, so that's really how PDS-0101 works. To date, we have reported preliminary and interim data from 3 of the 4 ongoing phase 2 trials. Our PDS Biotech sponsored program, the first one I'll walk through, is being performed in recurrent metastatic HPV positive head and neck cancer, for which the FDA-approved standard of care is the checkpoint inhibitor Keytruda. So you notice that for the first 2 trials, we're combining with standard of care. And the approach here with combining with standard of care is to rapidly get into the commercialization pathway. Oncologists, understandably, are very concerned about putting their patients on new drugs if they haven't been proven. So by combining with standard of care, these patients will still be getting the FDA-approved standard of care. However, if we are able to demonstrate substantial improvement in efficacy without compounding toxicity that comes from the approved standard of care, we believe that this provides us with a rapid commercialization pathway and strategy. So here, as we see in the first one, the preliminary data presented at the ASCO conference in June from our trial, which combines PDS-0101 with Keytruda, showed a 41% response rate. This means that 41% of these patients treated with PDS-0101 and Keytruda experienced significant tumor shrinkage of 30% or more. Right now, this compares favorably with the response rate reported for Keytruda monotherapy in this population, which is 19%. Right, so more than a 2-fold improvement over the reported efficacy rates for Keytruda monotherapy. Also very important was the survival of the patients on PDS-0101 plus Keytruda, which at 9 months was also very promising at 89%. And so with Keytruda, what's been reported in Keynote 048 study is that at about 12 months, there are approximately 49% of the patients alive. So again, very promising when we look at not just the objective responses, but also the potential durability of the combination in these patients. The second trial I have on here is the MD Anderson trial. Again, this is the MD Anderson Cancer Centers running a phase 2 trial in locally advanced cervical cancer patients. These are what they refer to as the high-risk patients. These are patients who have large tumors of 5 centimeters or larger. So here we're looking at a pretty high bar in terms of the size of the cancer. Many of these patients will also have had metastasis into the lymph nodes. So in this trial, again, PDS-0101 is given with the standard of care chemoradiotherapy. Now, the preliminary data in the first 9 patients, which I mentioned was presented at SITC this year, showed a 100% response rate. So every single one of these patients within a month had had tumor shrinkage of 60% or larger or more. Now, in 8 out of the 9 patients, they reported complete responses, meaning these patients no longer had any evidence of the cancer. So an 89% complete response rate, again, which is very compelling in this patient population. One of the big drawbacks of this standard of care is potential recurrence of the cancer. And so these patients are being monitored for over a 2-year period. And what we see here is that even up to 1 year, which many of these patients had been through, not a single one of these patients who had the responses saw any recurrence of the cancer. So again, a small patient population, they continue to generate more data, but very, very, very encouraging so far. For the third phase 2 trial, this is being led by the National Cancer Institute, and we have reported data from 2 arms of this trial. And so this is where we're combining our Versamune technology with our IL-12 technology platform, which IL-12 we refer to as PDS-0301, right? So here in this trial, we are combining PDS-0101 with PDS-0301 and a checkpoint inhibitor. As I mentioned, we reported data from 2 arms of the trial. This trial is being performed in advanced cancer patients. So 100% of these patients have failed chemotherapy treatment. 90% have failed radiation treatment, right? A portion of these patients have also failed subsequent treatment with checkpoint inhibitors such as Opdivo and Keytruda, right? These checkpoint inhibitor refractory patients have very few remaining options. Their historical rates with standard of care, historical response rates with standard of care are 5% or less. And their historical median survival is only about 3 to 4 months. So what you see here is that in these patients with our novel triple combination of PDS-0101, PDS-0301, and the checkpoint inhibitor, we have reported 21 months median overall survival among 29 HPV-16 positive patients. So 3 to 4 months, and now with the triple combination, we're seeing 21 months overall survival. And really to put this into perspective, if a patient was diagnosed this month as having failed checkpoint inhibitor therapy, on the average, they would live till maybe April. The patients on our drug, however, live to go through 1 Christmas, and the vast majority of them will see a second Christmas and counting. So very, very encouraging results to date, right? And I mentioned that the response rates in these patients with the standard of care is less than 5%. In our optimal dose group, the patients who got optimal doses of PDS-0101 and PDS-0301, we've seen a 63% objective response rate in these patients. So again, very encouraging, especially in a group of patients who have really no other alternatives, a significant unmet medical need today. Now, in the second arm, we're treating patients who, after failing chemotherapy and radiation treatment, would traditionally be moving on to checkpoint inhibitor therapy for the first time. So in these patients, when they go on to checkpoint inhibitor therapy, the reported response rates are less than 25%. And the reported median overall survival for these patients is also less than 12 months. Right, so 25% reported objective response rates and overall survival less than 12 months. In our phase 2 study, the National Cancer Institute has reported a response rate of 88% in this patient group. And at a median follow-up of 27 months, 75% of the patients remain alive. Again, we have seen not only encouraging early responses, but we've seen the durability of the immune responses, the generation of memory T cell responses, helping to stave off the cancer and continue to keep these patients alive for a substantial duration of time. So again, very encouraging results across all 3 trials. We've seen consistency in the data across multiple trials from multiple investigators, and some of these are the Versatile 002, for example, is being run at multiple sites within the United States. Again, very highly encouraging. with the Versatile-002 trial, I don't think I mentioned that earlier, we have been given fast track designation by the FDA. We have had initial discussions with the FDA, and so the FDA has given us the go-ahead to prepare and given us some very useful feedback on what they would like to see in a registrational trial. So we plan to move that trial into the registrational trial pathway approximately a year ahead of the planned schedule. So again, very encouraging for us, and we hope to get that registrational trial with Versatile-002 started sometime later this year.
Thank you for that summary. That's been, these results are really outstanding and are a tremendous improvement over the standard of care that we've seen. One of the things about it that struck me about them was that while checkpoint inhibitors can be very effective, the response rate is low, and you have a higher response rate and a durable effective action. So those are the two keys that I think are really distinguishing PDS from the other drugs that have attempted to improve the response rates in the checkpoint inhibitors.
No, that's an important point. The checkpoint inhibitors have been revolutionary in terms of treatment of cancer. In the patients in which checkpoint inhibitors work, you see very dramatic and durable responses. But those patients, as you mentioned, are far and few between, right? Because again, checkpoint inhibitors don't necessarily activate and prime a tumor-specific T-cell response. They more or less decamouflage the tumor and make it more visible to the immune system, but you still need to have those tumor-attacking and tumor-infiltrating T-cells available to then go in, attack, and kill those exposed tumor cells. And very importantly, I mentioned the memory T-cell response, right? In order to generate a continuous attack on those cancers, the immune system has to continue to remember what the enemy is, what the target is, and continue to do that and attack those cancers over a substantial duration of time in order to have that durability and long-term response against the cancer.
Some of the data that you presented was on the biomarkers and the things that would correlate and support the idea that the drug regimen is actually stimulating the T cells and the right type of T cells, and maybe even protecting against metastasis. Could you comment on that for a moment?
Yes, actually, that's a very, very good point and very good question. And that is where the studies that were done, the biomarker studies done both by MD Anderson Cancer Center and the National Cancer Institute are very important, because one of the key things that we have said as a company several times is that not all T cells are created equal, right? Simply generating a T cell response doesn't necessarily mean that it's going to translate into a clinically relevant immune response. You have to be able to generate the right type of killer T cell, right? It has to have the right killing potency, which is what we describe as a multifunctional killer T cell. And you have to be able to generate these in the patient's body in the right quantity, right? So you need the right type, you need the right potency, and you need the right quantity. If you can achieve all three, then there is the potential that it will translate into a clinically relevant response. And so when you look at the biomarker data that MD Anderson generated and presented, for example, they looked specifically in the tumors of these cervical cancer patients. And what they showed was that in these patients, we're generating accumulation of this right type of tumor targeting CD8 killer T cell, so that were multifunctional, that had high potency. What was also very interesting about the data they presented was they looked at what we call the circulating tumor DNA, which is really quantifying the tumor cells that are circulating in the patient's blood. And what they demonstrated was that after chemoradiotherapy occurred, even though you see the tumor shrink, there was actually a spike in the number of cancer cells circulating in the patient's blood. But as they showed the accumulation of these powerful killer T cells in the patient's tumors, as that increased, the circulating tumor DNA went down to 0. And there was a strong correlation between induction of those multifunctional killer T cells and the decline in the circulating tumor DNA. So it was very compelling data in multiple patients where this correlation was very clearly shown. And this was done in locally advanced cervical cancer patients. But when you look at the National Cancer Institute data, on the other hand, they looked at these very late stage terminal cancer patients who had failed all therapies, including checkpoint inhibitors. And they showed very similar responses immunologically in this patient population, where they also showed the induction of the multifunctional killer T cells. They showed very specifically induction of HPV-16 specific killer T cells. They show that 100% of the patients who responded had these HPV-16 killer T cells. And so very compelling data suggesting that even in these very late stage patients who have been on multiple therapies and who are very likely to have somewhat compromised immune systems, that even in these patients, we could generate these powerful multifunctional killer T cells that led to regression and shrinkage of tumors and long-term survival of these patients. So I think that was very important data that was generated by both parties.
Yeah, and one of the conclusions that I came to after reading that data was that all of these biomarkers support the concept of the HPV-16 antibody and the checkpoint inhibitor, and all
Right.
correlate with the patient observations that we're seeing in the survival data. So,
Exactly.
so it's not happening by chance. There's a real explanation and biomarkers that correlate with the effects that you're showing.
Right, and I think that was very important because even with some of the skepticism around the T cell activating technologies in cancer vaccines, well, how are these really working? It was very important and compelling to show that, okay, the PDS-0101 is doing exactly what it has been designed to do, which is recruit these T cells, prime them, activate them, and they actually do infiltrate the patient's tumors and have led to the results that we're seeing across multiple clinical trials.
Yes, and speaking of multiple clinical trials, you have a trial where you have patients who have exhausted all possibilities and are really at the end stage of the disease, short expectations for survival and no hope, and you've extended their lives. And then you also have a trial where the drug is added early as part of standard of care. So it's moving up in the treatment paradigm from patients who have nothing to lose to patients who can actually benefit and are at earlier stages. So it's going from a small end stage market to a potentially larger market. What would your strategy for phase 3 and regulatory approval be to fit these 4 trials together?
So I think our strategy now is, in terms of rapidly getting into the registrational pathway, we're focusing on 2 trials. The Versatile-002, for which we've been granted the fast track by the FDA, and the triple combination in patients who have really very few alternatives, who failed all options available to them. Now, the strategy here and why we're going with those 2 first is with the Versatile-002, we're going into a population who are a little bit earlier and who already have an FDA-approved standard of care, which is Keytruda, right? And so the strategy there is put PDS-0101 together with Keytruda, hopefully in the registrational trial, repeat the results we've seen here today, where we show significant enhancement of clinical benefit. But very interesting, we've shown no compounding of the toxicity due to Keytruda to date, right? What we've actually shown is a potential reduction in the toxicity of Keytruda, right? Keytruda in Keynote-048, they reported approximately 17% of the patients saw treatment-related grade 3 and higher toxicities. What we've seen to date in the first 43 patients who've gone through our independent drug data monitoring safety committee, not a single one of these patients has yet experienced any grade 3 or higher toxicity, right? So the good news here is that the combination does appear to be quite well tolerated. We don't see any compounding of toxicities. And so we believe that that is one where if we are able to replicate these results in the larger registrational trial, we see a pretty clear path to commercialization there, and hopefully ahead of the markets in terms of the first to get to into that stage. With the triple combination, on the other hand, that contains at least two novel compounds. We have PDS-0101, we have PDS-0301, which is the tumor targeting IL-12, and then we have the checkpoint inhibitor. And so there, we believe that the most rapid way to get there is to start in these patients who have failed all their treatment options. We see very compelling data here, where we're going from the historical 3 to 4 months to 21 months overall survival. And this actually, the 21 months includes both optimal and suboptimal doses of the drug, right? We are hoping to move forward with the optimal doses. But in these patients in the registrational trial, the goal there is to generate a substantial database where we have substantial safety information as well as efficacy and durability. And that, we believe that's the most rapid path to commercialization. We're hoping to have these discussions with the FDA this quarter, but then we're then able to move it earlier into the treatment paradigm, right? Where we then able to address checkpoint inhibitor naive patients, where again, the data is very compelling, almost 90% response rates, and also at 27 months, 75% of the patients still alive. But these patients have other options today, which are the checkpoint inhibitors. And so we believe the fastest way to go first with the patients who have no alternatives, get that established, and then move it forward. And so that's how we're approaching it to start.
Okay, and one of the things you mentioned was the triple combination and suboptimal doses. When you have a regimen with multiple drugs, the FDA usually wants to know what each drug is doing.
That is correct.
And do you have data to show that the role of all three drugs is contributing, or
Yes.
how to address those questions?
Yeah, so you're absolutely right. One of the key things that the FDA wants to understand is the role of each of the components in the combination, as well as the safety of each of the individual components and the safety of the combination. So what we have done is we have extensive preclinical data, which has actually been published, which shows that each of these drugs contributes towards the therapeutic outcomes. Very importantly, what it also shows is that PDS-0101 actually forms the anchor here. And so what we see is two synergies. You see a synergy between PDS-0101 and PDS-0301, which leads to what we call T-cell clonality, meaning that the T-cells become less diverse and more focused and targeted on the cancer. We see improved infiltration of these killer T-cells into the tumors and tumor shrinkage. Now, when you look at PDS-0101 and the checkpoint inhibitor, we see a very similar synergy. However, once you remove PDS-0101 from that combination, these synergies go away, right? And so it's very important, but when you have both of those synergies coexisting in the triple combination, you get the optimal anti-tumor responses. So in the preclinical studies, we have demonstrated with very compelling data the role of each of the three components in the eventual clinical outcomes. However, the FDA, we believe, would also want to see some clinical demonstration of the need for those three components. And so in the studies that have been done at the National Cancer Institute, we've talked about this optimal and suboptimal doses. That's one of the reasons why they're looking at optimal doses of, say, PDS-0301, for example. And what we show very clearly is that at the higher doses, you see significantly enhanced response rates as well as prolonged durability of the survival. So they clearly show the role of PDS-0301 in the combination. With PDS-0101, the biomarker data that we just talked about shows induction of these T cells, and very importantly, the HPV-16 specific T cells, which are directly related and correlate with clinical benefit. But what's also very interesting is that there were 7 patients who were recruited whose tumors were not HPV-16 specific. Those patients got the triple combination. and not a single one of those patients saw tumor shrinkage, right? And so again, that also really highlighted the critical role of PDS-0101 in the combination. With the checkpoint inhibitors have already been approved to address HPV-related cancer. So we are quite comfortable looking forward to those discussions, but we've shown both in the preclinical setting as well as in the clinical setting the role of each of these components in the clinical outcomes that we are seeing today.
Wow, that sounds like you've really anticipated this question and tested them not only in all combinations, but even the null hypothesis to prove that the HPV-16 positive tumors are the reason for the response.
That is correct.
Okay, so could you just discuss a little bit about Versamune as a platform and the pipeline indications that might follow?
Sure. So the Versamune technology, what Versamune does is Versamune is a lipid platform that can be combined with various tumor-specific proteins or fragments of those proteins. What Versamune then does is Versamune is able to present these proteins or these fragments of proteins into the right immunological pathways to really generate these, or to train or prime the T cells to recognize that protein as the foreign agent, and also activate the immune system, activate these T cells to then invade the body, seek out and attack the cells in the body which contain that unique protein, which in this case would be the cancer cells, right? So really that's how Versamune works. So Versamune forms the backbone of our pipeline by being able to take that tumor-specific protein, train the immune system and arm the immune system to then go out, seek and attack those tumor cells. What we have done is in our preclinical studies, we've gone beyond HPV. Our PDS-0102, for example, contains a protein that we call TARP or TARP. This protein is present in over 90% of prostate cancers and over 50% of breast cancers and 100% of acute myeloid leukemias. What we have demonstrated in our preclinical studies is that we can generate very similar levels of these multifunctional TARP-specific killer T cells. Right, and so the preclinical studies with PDS-0102 are complete, and we should hopefully be moving those into human clinical trials in the near future. PDS-0103, on the other hand, we're looking at a protein called MUC1, MUC1, which is present in a broad range of solid tumor types, including colon cancer, breast cancer, bladder cancer, ovarian cancer, and so forth. With PDS-0103, we have very similarly demonstrated that in preclinical studies, we generate very similar numbers of these multifunctional tumor-infiltrating killer T cells. PDS-0103 is now going through clinical manufacturing. We actually pushed PDS-0103 back a little bit, as we mentioned last quarter in our earnings call, to prioritize moving PDS-0101 into the commercialization pathway and registrational trials. However, we still do anticipate that sometime this year, we will be moving PDS-0103 into the human clinical trials, taking a very similar approach as we've done with PDS-0101, where in checkpoint inhibitor naive patients, we could potentially combine with either our PDS-0301 or a checkpoint inhibitor, and maybe in the checkpoint inhibitor refractory patient population, we could look at a very similar triple combination approach where we combine PDS-0103 with PDS-0301 and potentially a checkpoint inhibitor based upon the really promising results we've seen with PDS-0101. So I think the PDS-0101 program is a proof of concept program, right? We're looking at some very compelling indications, but it's really demonstrating how the technology works and how both PDS-0301 and Versamune really form a platform that we could very broadly apply beyond HPV-associated cancers.
Great. Okay, and just to summarize, could you go through some of the milestones that can be expected in the coming year, just in sequence or?
Yes.
by-
So we talked about the Versatile 002 trial. One of the key things that we would hopefully be able to get sometime during this year would be additional durability and overall survival data for this trial. We have not yet announced or presented any overall survival data for the trial. The good thing there is patients need to die to achieve a median overall survival. As of 9 months, we're still having very good survival. So that's protracted, but we hope to be able to at least provide an update on what the patient survival looks like in that trial. The goal is also to get this trial into the registrational trial pathway sometime this year. So that's also something that we could look forward to. With the triple combination, we are hopeful that we will be having a discussion meeting with the FDA this quarter. And so we'll hopefully be able to give some feedback on that meeting with the FDA and what the registrational trial pathway would look like for that triple combination. There are some key things that we would like to discuss with the FDA in terms of potential trial design. So that could, again, we'll have to make some decisions, but that could also potentially go into the registrational trial sometime this year. MD Anderson and the Mayo Clinic trial, we didn't talk about the Mayo Clinic trial today, but those are both investigator-initiated trials. So we have much less control over when data is released from those trials. But we are hopeful, as MD Anderson announced at SITC, that they had recruited 17 patients into the trial. We saw data from 9. And so we are hopeful that this year we'll also be able to hopefully get some additional data from that cervical cancer trial. And also with the Mayo Clinic trial, that's a trial in even earlier stage patients where it's a neoadjuvant treatment. These patients will get the treatment before they go into their robotic surgical removal of the tumors. And so Mayo Clinic, my understanding is that they'll be looking at very similar things to what MD Anderson looked at, for example, circulating tumor DNA, for example, shrinkage of the tumors. And so there is a potential that we could get some data from that trial too. But very importantly, moving beyond PDS-0101. As we just discussed, the hope is to get PDS-0301 also into human clinical trials later this year.
Great. That sounds like you have a lot of data and a lot of milestones coming up. So we'll look forward to following those. And I'd like to thank you, Dr. Bedu-Addo, for joining us today and sharing the fundamental progress and all of your views about PDS Biotech. Thank you very much.
No, thank you very much. It's been a pleasure, and I was pleased to participate in this event. Thank you very much.
With more than 6,000 small and micro-cap companies listed, if you're looking for the next Apple, then Channelchek truly is the orchard. It's an investor community where you can access independent Wall Street research, advanced market data, balanced news, corporate videos, and podcasts. You can even register to attend in-person Meet the Management Series luncheons and to be part of the preeminent annual Small and Micro-Cap Investor Conference, NobleCon. And here's the best news. Since Channelchek is sponsored by the featured companies, premium membership requires no investment. It's absolutely free. So check it out. Log in today. Find out why Channelchek is North America's leading source of research on the companies that just may change the world.